Now for our UV spectrometer results, for 555 we got 1.89, for 344 we got 1.88, for 308 we got 0.55, and for 406 we got 1.87. Now that means for most of them we had relatively uncontaminated DNA, except for 308, which was contaminated with proteins. Go on to, and then move on to amplicon PCR. Amplicon PCR, amplifying. God damn it. Then we move on to amplicon PCR, which amplified regions on the 16S ribosomal RNA gene. The amplifying the V3 and V4 regions. The PCR action was put through 25 cycles, producing 2 to the power of 25 copies. We included in this the negative and positive controls. After this, we put it through gel electrophoresis. This essentially let us know whether or not we had contaminated any of our products, meaning if our negative control had any DNA results in it, then that means that our DNA was contaminated. And if one of our results did not show up, that means that did not have any DNA. Now this happened by adding a phosphorescent, no, that's not true, I'm saying random stuff. This technique used charged particles in order to push this process used charged particles to migrate. Now over here, as you can see, we have our positive, our negative, our 344, our 555, our 406, and our 308. Now for our positive control, we did get a little line, which means that there was DNA in the sample. For our negative, there was not, which means we did not have DNA in the sample. So right now, we can see that our products were not contaminated with DNA. Now for our 4, 5, and 6, we did get results. For 8, we did not. So that means that for 308, we did not have any DNA that could be read. And then we move on to Ampere kit, PCR cleanup. And then we moved on to PCR cleanup and indexing PCR. For our first PCR cleanup, we added DNA. We bound the DNA to magnetic beads, and we used a magnetic stand to separate that from everything else, and then we used an ethanol wash to get rid of all the impurities. To index the PCR, we put PCR labels on the DNA, and then we'll only run 8 cycles. Whatever. And then after this, we put it through fluorescent spectrophotometry. After this, we put it through fluorescent spectrophotometry. This is more accurate than any of the other ways we have looked at before, and it determines the concentration of the amplified DNA. Whatever. After this, it was sent to a lab to be DNA sequenced using Illumina MISAC. After this, after we got our results back, each sample had a certain amount of reads, depending on which table had which results. For our sample 555, we had 10,129 reads. For sample 308, we had 20,034 reads. For sample 354, we had 14,862 reads. sample 406, we had 13,164 reads. Now, all of these were very, very high amounts of reads, so all of these had usable results. Now, when we go to our results, the highest result in each of the categories is as follows. For 555, the highest result in the phylum was Proteobacteria. In the order was Enterobacterials, and the genus was Enterobacter. In 344, the phylum was Firmicutes, order Clostridials, and genus Clostridium. For 308, it was Bacteroidals, and in order it was Bacteroidales, and in genus it was Provotella. In 406, the phylum was Firmicutes, order was Clostridials, and genus was Provotella. Alright. Alright. Let's just